Hi everyone, my name is Segun Oblugbode, and today I'll be talking about tissue regeneration of dental pulp. So, what are stem cells? Stem cells are special human cells that are able to develop into many different types of cells. These can range from muscle cells to brain cells, and in some cases, they can also fix damaged tissues. Currently, there has been a focus on stem cells and their possible application in science. So, how can these be used? They can be used to increase understanding of how diseases occur by watching stem cells mature into cells in the bones, heart, nerves. This has given researchers and doctors the ability to understand how diseases and conditions develop. Also, they can be used to generate healthy cells to replace disease cells, such as regenerative medicine. Stem cells can be guided into becoming specific cells that can be used to regenerate and repair disease or damaged tissues in people. So the focus of this presentation is to talk about how tissue regeneration can be used with dental pulp. Several studies have been carried out, and there is a promising niche with regenerative medicine and dental pulp. A well-known problem has been dental caries. What is dental caries? This is one of the most prevalent infectious diseases in the world, second to the most common one called cold. This is caused by poor oral hygiene, which causes dental caries. Current treatments such as chemotherapy, radiation therapy, and ingestions of medicine have been found to have some side effects, like excessive dryness of the oral cavity, and it affects salivation. The possibility of using exosomes isolated from odontogenic human dental pulp is what is the aim to use as biomimetic tools to affect linear specific differentiation. This can be used on primary human dental pulp stem cells, which is a DPSCs, and primary human marrow stromal cells, HMSCs, in vitro or vivo. Also, some studies have explored the possibility of achieving odontogenic differentiation of human periodontal ligament stem cells, which are PDLSCs, and human marrow stem cells for dental pulp tissue regeneration. So, some studies have explored using dental pulp as biomimetic tools for tissue regeneration. Exosomes are one of the core front runners up for this, and they are microvesicles that are secreted by cells to facilitate intercellular communication. There has been a renewed focus on their application in regenerative medicine. Recently, exosomes have been shown to increase the proliferability of mesenchymal and epithelial cells. A study carried out by University of Illinois, the Department of Oral Biology, hypothesized that exosomes isolated from differentiated cells could be used to induce lineage-specific differentiation of naive MMCs. Also, a second study by the University of France and the University of Illinois has identified stem cells from several dental tissues, has made dental pulp tissue generation a realistic clinical possibility, which included dental pulp stem cells and periodontal ligament-derived stem cells. So there were some various methods that were carried out during these studies, and specific methods were carried out by both studies to determine the possibilities for dental pulp regeneration, such as a cell culture of HMSCs. Using this study were cultured in minimum essential media, TM, which is a transmission electron microscopy, was used to verify the presence of exosomes in the purified samples, and also to evaluate binding of type 1 collagen. Fluorescence labeling was also used to identify the presence of exosomes, as well as binding experiments used to confirm certain binding proteins. For the second study, they also used an SEM, which is a scanning electron microscopy, for visualization. They used a alizarin red staining to visualize calcium depositions, and the use of dental pulp stem cell derived ECM scaffolds. So these are some results from the studies I just picked out that explains the successful identification and characterization of exosomes. So figure A to the top left shows the presence of exosomes in the isolates verified by TEM. These are electron micrographs showing the presence of exosomes, while figure C shows immunogold labeled the exosomes marker protein CD63. Figure C and D are proteins from the exosomes dental pulp stem cells, which were subjected to SDS page and immunoblotting for the markers protein CD63 and CD9. Representative immunoblots are presented to show the presence of both markers or both marker proteins. And the figure A to the bottom left represents a confocal micrograph of untreated dental pulp stem cells and pre-treated dental pulp stem cells with exosomes. They were subjected to exosomes endocytosis using fluorescence labeling. The exosomes in green in figure B at the bottom right shows a counter stained with tubulin antibody in red. This depicts the successful endocytosis of exosomes in the cell. And to the right top, this is a SEM imaging, which was performed to study the ultrastructural characteristics of cells grown within the dental pulp ECM scaffold. Results in the figure on the top right shows a pronounced cell matrix interaction of the 3 cell types with pulp ECM scaffold. The arrows in the figure point to cell processes interacting with the scaffolds, and collectively, these experiments demonstrate that DPSCs, PDLSCs, and HMSCs adhered, survived, proliferated, and interacted with their extracellular environment when cultured within the pulp ECM scaffold. And at the bottom to the right, this is a histology experiment, which shows calcium disposition within the control and ECM scaffold implant section, which were analyzed qualitatively using alizarin red staining. In this figure, this shows sections of the pulp ECM scaffolds containing DPSCs, PDLSCs, and HMSCs. And as we can see, a stronger alizarin red staining in the ECM scaffold containing all 3 cell types shows increased amount of calcium deposition. So for the discussion, I believe further studies are required to characterize the differences between earlier endocytosis and later signaling mechanisms triggered by exosomes, to understand the mechanisms behind the specificity of their actions. The studies did definitely hypothesize that along with cell-derived ECM incorporated biomaterials, exosomes can be used to recreate a complex extracellular environment that can enable a safe and reliable differentiation in stem cells. Also, the studies show that the biomimetic pulp ECM scaffolds can induce for an endotogenic differentiation of 3 different MFCs, namely the DPSCs, the PDLSCs, and the HMSCs. This study also proved that it is possible to achieve ECM-mediated linear specific differentiation of multiple somatic MSCs without the need for complex growth factor delivery systems. Thank you for listening. These are my references, and my name is Segun Oblugbode. Thank you.